In this video, Aurelia Biosciences Chief Scientific Officer Dr. Gary Allenby will be explaining his poster on screening for inhibition of protein-protein interactions in living cells, development of assays to detect modulation of proteins using nanobread technology.
Hi. So in this poster, what we're actually doing is that we're looking at protein-protein interaction, and specifically protein-protein interaction within living cells. So in this example, we're actually utilizing a technology from Promeda called nanobread. And what we're looking at is the interaction of two proteins, specifically ERK2 and a protein called SNORI3. So SNORI3 is implicated in bone formation, and our client wanted actually to look at the inhibition of the interaction between SNORI3 and ERK2. Okay, so what we're doing is we're basically doing a transient transfection assay in HEK293 cells using two plasmids, one of them containing SNORI3 and the other one containing ERK2. In this case, the SNORI3 was actually labelled with the nanolock enzyme, and the ERK2 was labelled with the fluorescent halo tag acceptor. So if I just draw this on the board for people. Okay, so what we have is we have our SNORI3, and we also have our ERK, and both of those were expressed in separate vectors, and both of those were then transfected into the cells. Yeah. And then basically then what happens is after 24 to 48 hours in culture, we have expression of our SNORI and ERK, but in this case our SNORI also contains on the end of it the nanolock enzyme. And on the end of our ERK we have a halo tag. Okay, so what we can do is by incubating the cells with a fluorescent ligand that binds to the halo tag, what we then end up with is we end up with a cell that is expressing both our SNORI3 and our ERK, and in this case the SNORI3 has a nanolock enzyme on the end of it, and in this case on the ERK we now have a fluorescent halo tag. Now what we can do is by expressing those in the same cell, we can then look to see if we get protein-protein interaction between SNORI and ERK, right? And we've got our nanolock enzyme on the end, and in the presence of substrate, in the presence of the addition of substrate, which is cell permeable, we then end up making photons of light, which are measured at 460 nanometers. And on the ERK we have our halo tag, which is actually our fluorescent tag. And when these come into close proximity, these photons are able then to activate this fluorescent tag and get it to produce photons at 600 plus nanometers. And then what we can do is we can then ratio the 460 nanometer signal against the 600 nanometer signal in order to look at if we've got something called nanobread, and BRET obviously stands for bioresonant energy transfer. In this case, the energy coming from these photons transferred onto this fluorescent ligand, which is then fluorescing, and we're able to detect that on a plate reader. In order to perform assay development and validation of this particular assay, we actually didn't have a compound that inhibited the interaction of ERK with SNORI3. And so what we had to do in order to validate it was actually to utilize another protein that would then compete for the binding of ERK. And what we did was we used the protein MEK2. And so what we're showing here in this figure is basically a competition assay where we've actually titrated in the protein MEK2 in order to see if we could remove ERK from SNORI, and then by doing so because ERK interacts with MEC, and then by doing so, what we would then end up doing is decreasing the signal because we decrease the amount of nanobreadth that we would have. And as you can see here, what we see is as we titrate in increasing levels of MEC plasmid, therefore MEC expression and MEC protein, we can see that our signal goes down. Over here, this actually shows a competition assay of a ERK inhibitor binding to ERK and preventing the binding of SNORI3 to it. And so in this case, what we've been able to do is we've been able to do a concentration response curve, shown here going from high concentration to low concentration. And basically you can demonstrate that as you increase the concentration of this compound, you can compete out the binding of ERK to SNORI. And in this case, with this particular compound, we had an EC50 around about 60 nanomolar. Okay, so the client wanted us to screen 20,000 compounds through this protein-protein interaction assay, looking for inhibitors of SNORI-ERK. And what you can see in figures 4, 5, and 6 is actually the validation and then the screening of the assay. So figure 4 shows actually the distribution of the compounds and the actives in this particular assay. So the assay ran really well, had good Z factors, and the distribution that we can see is mostly centered around 0, which is what we typically see. And if we set a 30% cutoff for the assay, we actually ended up with 1.3% hits, which is approximately 233 compounds. The Z factors, which are shown here throughout the course of the screen involved 56 384 plates. Z factors came out around about an average of 0.65 to 0.7, shown here. And lastly, when we went on to retest the 233 compounds, we got a really good retest between testing it in one assay and then testing it, repeating it in the same assay on a separate plate. And as you can see, we get a good line of correlation with our hits. And so on that basis, we then went on to pick particular hits that the client was interested in order to look at them against their selectivity specificity assays. So following on from the high-throughput screening, the identification of 233 actives, the client wanted us to test a number of these actives that they selected based on other parameters such as synthetic chemistry and drug-like properties. And so we went on, first of all, to look at the compounds in terms of a dose-response curve in the SNORI-ERK assay. And then we went on to look at selectivity, and finally we went on to look at what I call compounds which would affect the technology readout. So in figure 7, what we're demonstrating here is that for a number of the compounds we can get a dose-response curve. Some of them are more potent than others, but certainly you can get a nice sigmoidal curve dose-response. Figure 8 basically demonstrates selectivity. So in this case, we're looking at SNORI3, but actually there was another target which we didn't want to hit, which was actually SNORI2. And so what we're doing here is that we're looking at SNORI3 against the ERK interaction in an identical PPI assay, but just on a different target. And lastly, figure 9 actually looks at what I said before, which are technology, compounds that affect the technology. So in this case, we took a completely different assay from Promega. In this case, it was P53MDM2, which is another protein-protein interaction assay. And we ran the hits through that assay in order to see if any of the hits hit the technology rather than hitting the target. And there were a few compounds that actually affected the readout. So in summary and conclusion then, what we were able to do was we were able to demonstrate that we could build a PPI assay, in this case for SNORI-ERK, ERK2, SNORI3, And then in addition to that, what we were able to do is that we were able to screen 20,000 compounds through the assay. We were able to identify a number of compounds that actually inhibited that interaction of those two proteins. We were able to then test them for dose response, able to identify some which has reasonable potency. We went on to have a look at whether they were selective for Schnorr-3 by having a look at a PPI assay that involves Schnorr-2, and in addition to that we went on to look at a completely non-related assay, P53MDM2, in order to see if any of the compounds interacted with those. And basically we would suggest that this PPI assay format, which is available from Promega, is a really interesting technology readout in order to look at proteins in their natural habitat within the cell and to look at their interaction between each other and to try and identify compounds that inhibit that interaction.
Contact us if you want a copy of this poster or if you would like to learn more about our experience with protein-protein interactions.